Hi everyone, my name is Jessica Woodall, and I am a Chesapeake Conservation Corps member stationed at the Maryland Department of Natural Resources. Today I'm going to be talking about results from recent surveys and capture-recapture monitoring of dwarf wedge mussel in Nanjemoy Creek, Maryland. This is the capstone project that I am leading as a member of the CCC. This work is being done in collaboration with both Matt Ashton and James McCann of Maryland DNR, as well as the Chesapeake Conservation Corps. The dwarf wedge mussel is a small freshwater mussel that lives on sand and gravel bottoms of creeks and streams. It is federally and state listed as endangered and has been since 1990. It can be found in streams from New Brunswick to North Carolina. This species has declined significantly in the last 100 years due to industrial, agricultural, and commercial development, as well as water pollution, invasive species, and host fish deficiencies. This has left many populations fragmented and at low densities. Some recovery actions for the dwarf wedge mussel include developing habitat protection strategies, supporting propagation efforts, developing standardized survey protocols, and developing models to better understand populations. The dwarf wedge mussel was historically found in 11 streams and 7 watersheds in Maryland. Now it is found in only 4 streams and 4 watersheds located on the eastern shore and in southern Maryland. The population in Nanjemoy Creek is where this study is being conducted. Nanjemoy Creek is located in Charles County, and you can see the watershed circled on the map. The population of dwarf wedge mussel here is among the larger populations, which is likely because the watershed has been protected by land purchasing. In the summer of 2020, DNR conducted some dwarf wedge mussel surveys in Nanjemoy Creek. They used timed visual surveys conducted by either snorkeling or bucket searching. There are 2 streams in this watershed, Nanjemoy Creek and Beaver Dam Creek. The Nanjemoy Creek sites were 10 meters long, and the Beaver Dam Creek sites were 30 meters long. When a dwarf wedge mussel was found, a marker was placed at its exact location, and the mussel was placed in a flow-through container in the order of collection. All of the dwarf wedge mussels were externally tagged with shellfish tags and 10mm pit tags. Then they were returned to their exact location. During the initial surveys, 19 sites were searched, amounting to 60.32 person-hours. 4,500 mussels were found, with a majority being Elliptiocomplanata. 161 dwarf wedge mussels were found across 14 sites. The histogram shows the size distribution of the 4 sites I'm focusing on for my project. They are referred to as Beaver Dam Creek 2, Beaver Dam Creek 3, Nanjemoy Creek 3A, and Nanjemoy Creek 3B. Shell length varies at these 4 sites from 22 to 46 millimeters. Overall, the average shell length for all of the dwarf wedge mussels found was 31.5 millimeters. The ratio of male to female dwarf wedge mussels was fairly split, but there were slightly more females than males. There were also several unknown individuals. The results of DNR surveys showed that there were viable populations of dwarf wedge mussel in Nanjemoy Creek. We decided to develop a survey design to monitor them, because the data obtained from this work can be used to advance conservation efforts. We used a mark recapture design for 4 sites, with 2 in each respective stream. The sites differed slightly in size and the number of dwarf wedge mussel present. For example, Beaver Dam Creek had 20 and 43 dwarf wedge mussels, while Nanjemoy Creek had 10 and 17. During the monitoring events, the sites were scanned with a submersible pit tag reader for a set amount of time, depending on the site length. The 30-meter sites were scanned for 30 minutes, and the 10-meter sites were scanned for 10 minutes. There was also an additional 2 to 3 minutes of downstream scanning to identify any tagged mussels that moved out of the site. We used a robust design for this survey. This includes 3 open sampling periods about 3 months apart. Within each primary sampling period, or in our case, each season, there are 4 closed sampling periods about 2 weeks apart. These are the monitoring events. During each monitoring event, discharge was measured in each stream and temperature was taken at each site. The data collected from the events will be used to model survival and detection in the program MARC. After the last primary period, another visual survey will be conducted to measure growth of the tagged mussels and to identify any new mussels at the sites. Here are some figures showing the data collected during the first primary period. The figure on the left shows the proportion of mussels recovered over time. There is a general decline from T0 to T2 by about 0.5 or more, but more mussels were found in T3 and T4 compared to T2. The overall trend is that there were fewer mussels recovered over time, but it was variable from event to event. The figure on the right shows the discharge for each stream over time. This shows that discharge did increase over time for both of the streams. When comparing the two figures, there is no clear trend in mussel recovery with the stream flow. This slide also shows the proportion recovered figure on the left, but the figure on the right shows the stream temperature over time. There are a lot of gaps in the stream temperature data, but you can see a decrease in stream temperature from the second to the fourth events. When comparing these two figures, there is also no trend in mussel recovery with the stream temperature. Because recovery was high at the end of the first primary period, in spite of increasing flows and decreasing temperature, we assessed apparent survival with the live recapture Cormack-Jolly-Sieber model. The two most likely models included capture probability dependent on time with constant and time-dependent survival. Apparent survival estimates for the time-dependent model varied from 0.88 to 0.99, and detection from 0.63 to 0.88. The next steps for this project are that we will continue pit tag monitoring into the third primary period, and we have already started conducting the monitoring events for the spring. In the summer, we will resurvey the sites to get growth estimates for the previously tagged mussels and to identify and tag any new mussels. With the completion of all of the events, we will run both open and closed population models under the robust design to estimate apparent survival and detection probability. This project will continue on with DNR after this initial run, and it will be adapted as needed. Finally, I would like to acknowledge the wonderful people and organizations that have helped me on this project, including Jackie Savalia of Maryland DNR and several Chesapeake Conservation Corps members. I would also like to acknowledge DNR's Wildlife and Heritage Service, as well as the U.S. Fish and Wildlife Service Chesapeake Bay office for their support on this project as well. Thank you.